Hi everyone, so in this video I'll talk about protein engineering. So let's get started. So starting with a small introduction about what is protein engineering. So one of the most exciting aspects of recombinant DNA technology is that it permits the design, the development and isolation of proteins with improved operating characteristics and even completely novel proteins. So some of the common properties that need to be modified, which is the stability, the binding affinity, catalytic activity, as well as the substrate specificity. So as we know, some of the protein features or some of the protein molecules do not imbibe some of the characteristics that is very much needed. So we need to modify those protein molecules accordingly so that that particular protein molecule possess that particular characteristics or particular feature. So there are some of the important modifications, as you can see, such as the substrate, the catalytic, binding and the stability affinity that needs to be improved for the increasement or enhancement of a protein molecule. So moving with that, so talking about the methods of protein engineering, so we'll be following two main methods of protein engineering and how it will be carried out. So one of the method is rational design, and next we have the directed evolution or combinatorial design. So these are the two methods that we are going to talk about protein engineering. So you can see here the difference between the two as well here. So this is the directed evolution and this is the rational design. So rational design includes the wild type or the parent gene, alright, whereas the directed evolution includes the additional parent gene. And we have some of the important features about rational design is it consists of structural or structure analysis modeling, we have site directed mutagenesis, we have ligation and transformation, and then we have protein expression and characterization. So these are the some of the points about rational design, whereas the directed evolution consists of random mutagenesis and not site directed mutagenesis. So it consists of random mutagenesis, then we have gene library ligation, which is very much similar to the rational design, then we have protein library expression, and lastly we have the screening and the evolved variant, which is this at last we get. So this is the kind of modification that is carried out using protein engineering. So these are the two main methods that are used during protein engineering. So moving with that, so talking about the rational design, so talking about some understanding more about rational design. So rational alteration of selected residues in a protein to cause predicted changes in function. So it requires detailed knowledge of enzyme structure, function and catalytic mechanism. So one of the most important points about rational design is we need to have the knowledge of enzyme structure, the function, the catalytic mechanism, whereas it is not the same for directed evolution. For directed evolution we do not need to know so much, whereas for rational design we need to know everything about that particular molecule, so whether it be structure, its functionality or its mechanism. So after we know everything, we can perform changes accordingly. So some of the examples that include rational designs are the rational design of tyrosine transfer RNA synthetase and B-lactamase demonstrated the power of this approach in understanding enzyme catalysis and engineering enzyme activity and specificity. So some of the examples or some of the very old designs of tyrosine transfer RNA synthase and B-lactamase was done to demonstrate how this thing works. So we have some of more, such as the design of T4 lysozyme with increased stability by engineering disulfide bonds into the protein. So by putting some more disulfide bonds into T4 lysosome, its stability has increased. Alright, so to increase the stability, these are some of the methods the scientists have incorporated over the years to increase the specificity or the characteristic feature for particular molecule. So we have another example of the redesign of a lactate dehydrogenase into malate dehydrogenase by introducing a single point mutation. Alright, so the redesign has been done of lactate dehydrogenase into malate dehydrogenase by introducing a single point mutation. And lastly we have the design of subtilism enzymes with increased activity and altered pH profiles by engineering surface charges. So there are a number of methods they have incorporated to bring about changes in these molecules. Let's say it be by single point mutation, it be by inserting disulfide bonds or engineering surface charges, and there are many other methods in rational engineering. But before that, we need to know about that particular molecule, its functionality and its catalytic mechanism to perform these rational design. So moving with that, so some of the rational design techniques, or the two main rational design techniques that is followed, is the site directed mutagenesis and we have the domain swapping. So these are the two main important features or important rational design techniques that are followed. One is the site directed and we have the domain swapping. So in site directed, it's very simple. So in this particular site directed mutagenesis, it's a molecular process in which intentional changes are made at particular locations. Alright, so let's say this is a DNA sequence. You can see 5 to 3 and 3 to 5. The template is also present. So we want to ligate this part. We want to restrict that or particular cut this region at this site. So you can see two different regions or two different parts of a single DNA sequence we can see. So this is what happens in site directed mutagenesis. We can bring about some specific changes by site directed, or it can bring about changes which we require actually. So in mutagenesis, we never know which particular DNA sequence or which particular location. So let's say this is the DNA sequence. We do not know which particular nucleotide. So there are lots of nucleotides, as we know. So which particular region of this nucleotide will mutate? Alright, so whether it can be this or this or this. So in mutations, we never know. In random mutations, we never know the location about this. Whereas in site directed mutagenesis, we can control that. If we want to mutate this particular part, or we want to ligate this part, or whatever we want, we can do on our wish. So it's pretty much on our hand under site directed mutagenesis, and intentional changes can be made. So this is how it's done. We can produce a cut here through PCR, and thereby we can again ligate it by overlap extension PCR. So these are some of the important methods regarding site directed mutagenesis. Talking about the domain swapping. So although the site directed mutagenesis is effective in altering protein functions, it often results in incremental improvement of protein functions rather than dramatically improved or novel protein function, because it can only modify protein sequences at single or multiple sites. So it definitely, it's a plus point using site directed mutagenesis, because it is definitely altering the protein functions. Also, it's improving the protein functionality. Also it is expected that the creation of novel protein functions may require dramatic changes in protein structures, and to address this limitation, one simple genetic method to introduce novel protein function is to combine protein domains from different proteins, or so-called domain swapping. So the problem here with site directed mutagenesis lies, so that we are using domain swapping, is that so it is expected that the creation of novel protein functions may require dramatic changes. So it brings about some of the dramatic changes in the novel protein structure, which we do not actually require. So whenever we particularly try to mutate certain region or modify the function of a particular protein region, so it dramatically improves the novel protein function, which we do not want. So in that case, domain swapping is used. So this is another technique of rational design, in which a protein domain is typically defined as a folding or a functional unit of a protein, and because many large protein contains several functional domains, and the fusion of domains with different functions may result in a multifunctional protein with unique features. So what happens in domain swapping? We just swap the domains with a particular region. So we just remove a particular protein domain with another protein domain, so as to increase the specificity, so as to increase the functionality of that particular part, so that the overall dramatic change in a novel protein does not occur. So we do not want the overall dramatic change, we just want the particular site to have that or to own that particular change. We do not want the entire thing. So for that purpose, domain swapping is done. So moving with that, talking about the drawbacks of this design. So the real truth is that our current ability of rational design protein is still rather limited. It is because apparent that owing to the intricate and complex relationship between protein structure and function. So it's not. It's also. It's. I would say that it's still under experimentation. It's not completely proven, but it's successful to a point. Also, rationally introduced mutation often have unexpected disastrous effects on enzyme stability and activity. So, as said, these are not actually 100% proven, so these are under experimentation. Also, so thus to circumvent the unpredictable effects of individual residues, methods that are able to access mutation at single sites or multiple sites in a large scale format were well developed in the 1990s. So it was long before that these were developed. Also, this newly trend ultimately led to the establishment of directed evolution as a powerful approach for protein design. So it led to the another method of protein engineering, which is directed evolution. So coming to directed evolution with this. So on the directed evolution, we have the gene is mutated either at a discrete site or random, and then selection is made for protein variant with the desired property. So also the improved variant can be subjected to further rounds of mutagenesis and selection known as directed evolution. So under directed evolution, the particular gene that we want to mutate is either at discrete or at random position. So it's not very much specific as we observed at the rational design. So in this, selection made for protein variant with the desired properties is done at random sites. Also, the paradigm for this approach is the enzyme subtilism. So every property of the serine protease has been altered, including its rate of catalysis, substrate specificity, pH profile rate, stability to oxidative, thermal and alkaline inactivation. Also, variants also have been produced that favor aminolysis over hydrolysis in aqueous solvents. So there are many other molecules that are used under directed evolutions, but which are very random, and also these are done in a way such that the overall specificity of the molecule is increased. So let's just keep this video till here. Hope you enjoyed this video, and thanks for watching.